All right, hello and welcome to my project on sirolimus, also known as rapamycin. Okay, so this drug inhibits the mTOR signaling pathway, and this signaling pathway is responsible for regulating cell division, cell metabolism, apoptosis, and cell death, as well as immune cell differentiation and the ability of these immune cells to respond to cytokines, interleukins, antibodies, etc., within the body. So that just constitutes the general inflammatory response. It's also known for being extremely overactive in many forms of cancer. So that's one of the reasons why this signaling pathway is of interest to cancer researchers. Now, as mentioned previously, mTOR is overactive in many forms of cancer. For example, 70% of ovarian and breast cancers have too much mTOR expression. AKT1 overexpression, which is a downstream protein in the mTOR pathway, is also found in 15% of melanomas. mTOR is also linked to many lung cancers. For example, 90% of, I'm gonna butcher this, adenocarcinomas and 40% of squamous cell carcinomas are also found to have abnormal mTOR activation. Now, my drug, sirolimus, is just the generic non-name brand form of rapamycin and Rapamune. I actually believe that Rapamune is owned by Pfizer, which you might know from the COVID vaccine. So sirolimus is an immunosuppressant commonly used in organ transplants, especially kidney transplants. I've already stated this, but basically what it does is it stops the recruitment of immune cells to a site of inflammation, which is invaluable in an immunocompromised organ recipient who obviously their own body is going to go into what is an autoimmune response targeting their new organ. Sirolimus is also used in the treatment of epithelial cancers, squamous cell cancers, etc., basically any cancer of a fast-growing cell. It's used in the treatment of lymphangiomyomatosis. It's also used to treat arthritis and osteoporosis. Okay, so getting into the mechanism of the mTOR pathway, we first start off with the fact that mTOR consists of two complexes, mTOR C1 and mTOR C2. Both of these complexes are activated by a ligand, in this case, a growth factor, binding to an RTK receptor and sending the signal down to this complex. mTOR C2 is activated by growth factors, which then in turn activates an AKT protein, which goes on to inhibit apoptosis, activate PKC, and increase the cell's metabolism. And the latter two of those effects will have the the end result of the cell dividing and proliferating. In cancer cells, this tends to be upregulated, leading to mass proliferation. However, rapamycin, and my drug also, the generic one, actually doesn't bind to mTOR C2, it binds to the mTOR C1 complex as an inhibitory molecule. So the mTOR C1 complex is located on the lysosomal membrane, and this thing is activated by two factors. First off, growth factors inhibit AMPK, which in turn activates TSCL2. And insulin, when it gets into the cell, activates AKT, this molecule, which in turn will start inhibiting TSCL2. So once TSCL2 has been inhibited, it will no longer inhibit REB, which allows for the activation of mTOR C1-2. So that's just kind of the upstream cascade of molecules from the growth factor to the mTOR C1 complex. Now, after this mTORC1 complex has been activated, it's first going to phosphorylate something called P70S6K, and this is an amplification step. This P70 is then going to phosphorylate CAD, which again is an amplification step, which then allows CAD to start activating and synthesizing pyridamine. Pyridamine, there we go. So that promotes that synthesis, which is again a necessary function for cellular growth. P70 also phosphorylates EF2K, which in turn fails to phosphorylate EF2, which means that EF2 is free to activate ribosomal translation of proteins and polypeptides. P70 also activates a protein complex called ELF4B, which also helps to activate the ribosome. And the P70 activation of other or phosphorylation of other complexes is an amplification step as well. So now these ribosomes, which are the 6S ribosomes or the S6 ribosomes, are now hyperactive in basically producing a lot of proteins and polypeptides, which in turn leads to cell growth and cell division at a massive rate. Okay, so lastly, mTOR also activates these two compounds, TFEB and ULK1, which it phosphorylates them, and it eventually leads to autophagy of old, no longer needed parts within the cell, which basically allows the cell to kind of eat its unwanted parts and, again, to get more energy and proteins and whatnot to use in cell division. So in short, the mTOR pathway is just very, very important in cell division. All right, and as you can see, I really only kind of went over this at the bottom here. This mTOR pathway is actually very, very extensive. So I just kind of covered the portions of it that were directly related to and inhibited by my drug of choice. You can also see here at the bottom that lipid synthesis, lipogenesis, et cetera, all of these functions are again related directly to cell proliferation and cell growth. So this is just activating those. Okay, so now moving on to my mechanism of action. All right. Why is my screen blank? Oh, there we go. Okay, so this study by Brown et al. From 1995, back when my drug was first being discovered and synthesized, I actually believe it was from a soil microbe on the Easter Islands that this drug was discovered in originally. That was a rapamycin. Okay, so basically this, the researchers Brown et al. Looked at how P70S6 kinase activity was affected by rapamycin. So how they did this is that they administered rapamycin to several tumor lines. They had a leukemia cell line, a melancarcinoma cell line, and a colon cancer cell line that they looked at. And they additionally had a mutant FRAP domain in vivo that is basically, it doesn't allow rapamycin to bind to its target complex. So what they concluded from this experiment was that rapamycin was determined to inhibit P70SK6 activity, making it a regulatory inhibitor of this pathway. It inhibited the S6 ribosomal function, which if you remember from my mechanism of action, my pathway, this is the mechanism of action. If you remember from my pathway, the S6 ribosome was the ribosome directly activated by mTORC1, which in turn was the primary propagator of cell proliferation and cell component synthesis. So rapamycin was found to inhibit S6 ribosomal function within tumor cell lines, and additionally, the FRAP complex without rapamycin autophosphorylated, meaning that rapamycin was an inhibitor of a working FRAP complex. That was not seen in the mutant FRAP complex. All right, and this study is actually insanely interesting to me because it looks at a more recent use of rapamycin to target mTOR complexes in neurodegenerative diseases in humans. So mTOR has actually recently been found to be abnormally regulated in many neurodegenerative diseases. These are things like all forms of dementia, Huntington's, Parkinson's, Alzheimer's, and ALS, stuff like that. So we already know that rapamycin inhibits the mTOR C1 complex by binding to FK506 and basically creating a stable, non-working mTOR C1 complex kinase. So what they did in this study was they looked at wild type in Alzheimer's disease modeled mice, which are mice that have genetic mutations that basically make their brains look similar to that of a human with dementia or Alzheimer's disease. And interestingly, what they found in dosing these two types of mice with rapamycin, was that normal synaptic function, the wild type mice, were actually incredibly debilitated by rapamycin. However, in the AD model mice, the neural cells were actually protected by the rapamycin. So not only did this help the mice with their not their short term, their long term memory, but it also was found to increase neuroplasticity and in some cases may have been attributed to a longer lifespan within these mice as well, which is incredibly interesting. So what they concluded from this is that rapamycin may potentially be a treatment for neurodegenerative diseases in cases where where inhibiting mTOR activity is beneficial. This obviously isn't in all degenerative diseases. However, it is in a lot of big ones, such as Parkinson's, which we don't really have a good therapy for currently. However, an unfortunate fact of rapamycin is that it can have some pretty bad side effects on humans. For example, the main one was actually extremely poor wound healing. So obviously, if you have an elderly person, they're already immunocompromised, they're already more at risk of severe illness just to being old, severe injury, just because their body's not what it used to be. So this isn't something that you'd want to take. You wouldn't want to take their ability to heal their own wounds from them, like whatever ability they have left. So that could be a potential limitation on rapamycin's usefulness as a target in neurodegeneration or a therapy in neurodegeneration. Okay, so in conclusion, my drug inhibits the mTOR pathway in order to stop cell proliferation and stop the immune inflammatory response by stopping the recruitment of macrophages and whatnot to the inflammatory site, to the site of inflammation. It's also an important cancer therapeutic for reasons I've already covered and is a useful immunosuppressant. Now, some future directions that this drug could potentially take is that it could be a potential therapeutic in neurodegenerative diseases such as ALS, Alzheimer's, Parkinson's, and Huntington's disease. And it also could be utilized as a therapeutic agent in cancer treatments where the cancer is a direct result of overactive mTOR activity within the proliferating cells. All right. So here are my works cited. Thank you very much for coming to see my my thing. Thank you.